Hello everyone, myself Rekha DN from the Department of Allied Science, RLJIT, Durbalapur. Today I am giving a lecturing on electron microscope. This is my third video on the material characterization. So in the last video we have studied the X-ray diffraction and Bragg's law. So now in this video I am going to explaining the electron microscope. So the topics that we are covering in this video, that is introduction to electron microscope, next the types of electron microscope, the principle of electron microscope, construction and working of electron microscope, advantages and disadvantages of electron microscope and its application. So before going to see the introduction of electron microscope, first I will tell you what we are using and what is the electron microscope is used for. So if I consider an electron microscope, so microscope we will use to magnify the size of an image or size of an specimen. So in this electron microscope we are using, instead of a light, an electron to use it instead of light. It is used for those objects which are smaller than 0.2 micron meter. So if I consider the history of this electron microscope, before going to see this electron microscope, scientists primarily used an optical microscope. This microscope uses a visible light to illuminate and to magnify specimens with limitation in resolution and magnification compared to electron microscope. These were the standard tools for viewing microscopic details before the electron microscope. They use lenses to focus visible light and magnify images of specimen. But in the electron microscope, scientists used a beam of electrons instead of light, allowing for much higher magnification and resolution, revealing details at the nanometer scale. Now, the first electron microscope was invented by a scientist, that is Ernst Ruska. He is a physicist, English physicist, in the year 1933, and he awarded a Nobel Prize for the invention of this electron microscope in the year 1986. So now, after using this electron microscope, so we can identify the difference between the optical microscope and the electron microscope. So in the given slide, see here the image from the optical microscope. So it is blur, it is not clear, but the image what we are getting from the electron microscope, it is clear. So this is magnifying and also high resolution image we are getting from an electron microscope. After studying the history of this electron microscope, let us explain the principle of the electron microscope. So if I consider the principle of electron microscope, it involves accelerating electrons and focusing them onto a sample using a electromagnetic lenses. Interaction between the electron and the specimen generates a signal that are used to create an image. So now in the given diagram in this slide, so we have a specimen and an incidenting electron beam. Now how to prepare this electron beam to incident on the specimen means, so in an evacuated chamber, so we incidentium we are kept an tungsten filament. So if I heat the tungsten filament, the tungsten filament, due to thermionic emission, it will emit a number of electrons. So that number of electrons emitting, which emits in all the direction, so that emitted electrons, it was making to incident in a one direction. So if I make that to travel in one direction, then it will become an high energetic beam. So this high energetic beam, when I going to incidenting on a specimen, some amount of electrons from the incidenting electron, so it is absorbed by specimen. Next, that incidenting electron beam will undergo interaction with electrons in the specimen, and that electrons will emit a number of electrons. When it is in phase, that as I told you in the lecture 2 second video, that emitted or the reflected electrons, if it is in phase, then it will form a one reflected beam. That beam, when it will exist, means when emitted electrons, if it is in phase, then it will create an electron beam, energetic electron beam. That beam we are calling as a X-rays. Now whenever you, this is one case. Next, if I consider the second case, that is backscattered electrons. So this backscattered electrons, if I consider that electrons, whenever the incident beam, when it is incidenting on a, whenever incidenting on a specimen, so then the electron will undergo, the incidenting electron will undergo again back from that specimen. So because that each specimen, inside the specimen, that number of atoms, ions are there, so due to that some electrons will scatter back from that specimen. So this electrons we are calling as a backscattered electrons. Next, if I consider the secondary electrons, so whenever I incident an electron on the specimen, so here the electrons whatever we have on the plate or then the specimen, so that specimen which has a number of atoms with a number of electrons, so that electrons will undergo emission. That emission we can call as a secondary emission. So that emission, that electrons we are calling as a secondary electrons. So after this, finally which kind of electrons we have, means that is transmitted electrons. So what is mean by the transmitted electrons, means so that is whatever we are going to incidenting an electron beam, so few of them will observe and few of them will undergo secondary electrons and few of them will undergo backscattered electrons and X-rays. So now next, the transmitted electron also we can see. The transmitted electrons, what we are getting, so from that transmitted electron, so for this transmitted electron microscope, from that only we are getting an image. So this is the principle of the electron microscope. So after this principle, next I am considering a transmitted electron beam. So this construction of the transmitted electron beam, what it consists of, means it consists of a the electron gun initially, and after this it consists of the magnetic condensing lens. Magnetic condensing lens, and why we are using is, means it is used to condense the electron and is also used to adjust the size of the electron that falls on the specimen. So the specimen placed in between the condensing lens and the objective lens. So magnetic objective lens is used to block the high angle diffracted beam from the specimen. And aperture, why we are using means this aperture will eliminate the diffracted beam. So why we are eliminating this diffracted beam means so because to avoid or to get the to increase the contrast of the image. So next after that we are kept a projector, so that is magnetic projector lens we are placed after the aperture. So this where we are kept means in order be to get the fluorescent screen. So in front of the fluorescent screen we are kept a projector, so where that we are getting a high magnified image. So this is the TEM, that is transmitted electron microscope. If I consider the working part of this, so according to diagram we are explaining here, so it this starting point electron source is there, so that is which emits the number of electron. The stream of electrons are produced by the electron gun and is made to fall over the specimen using the magnetic condensing lens. Based on the angle of incidence, the beam is partially transmitted and partially diffracted. Both these beams are recombined at the sphere to form the image. The combined image is called the phase contrast image. In order to increase the intensity and the contrast image, an amplitude contrast has to be obtained. This can be achieved only by using the transmitting beam, and thus the diffracted beam can eliminated. So now in order to eliminate that diffracted beam, the resultant beam is placed through the magnetic objective lens and the aperture. The aperture is adjusted in such a way that the diffracted beam is eliminated. So in the previous slide I told you that so we are eliminating a diffracted beam. So why means because to increase the contrast of the image. So next after that, so after this aperture we are kept a projector lens. Why means because for further magnification in front of the projector we are kept a photographic screen. So on that photographic screen we are getting a contrast magnified image, bright field image. So this is the transmitted electron microscope. So after this next we have a the scanning electron microscope, and one more in this transmitted electron microscope, so we can get a the image on the fluorescent screen, so that is black and white screen. And one more, so in this transmitted electron microscope, so the image what we are getting which gives the information about the structure of the specimen and also the internal structures of the material of it is in the range that is in nanometer range. After this let us see the next, that is construction of SEM. SEM means that is scanning electron microscope. It consists of an electron gun. See in the diagram, first we have an electron beam. Electron gun we are kept to produce high energy electron beam. How we can make it is into a beam? By keeping in front of the gun, electron gun, by keeping a positively charged anode, positively potentialed anode. So that is the steps. So after keeping the anode, the a magnetic condensing lens is used to condense the electron beam, and a scanning coil is arranged in between the magnetic condensing lens and the sample. So this scanning coil what we are using, it is scanning, it's like a TV scanner. So the electron detector is used to collect the secondary electrons, as I told you in the principle of the electron microscope. So whenever you incident an electron beam on the specimen, there are number of scatterings or number of electrons we are seeing. So like absorption of electron, next the backscattered electron, next the secondary electron, transmitted electron, and the x-rays, like number of electrons we are seeing. And in this SEM, so we are collecting secondary electrons. So that second, to detect that secondary electron, we are kept a detector. Why? Because to collect the secondary electron and can be converted into electrical signal. By collecting that electrical signal, so we can get a video amplifier in the cathode ray oscilloscope. This is the construction. So now after seeing this construction, let us see how it will works. So working of scanning electron microscope. So in this diagram, so whatever the operators we are getting, then let us see how all these operators are working in this SEM. So firstly, we are kept an electron gun or an electron source. So stream of electrons are produced by the electron gun, and these primary electrons are accelerated by the grid and anode. So already I told that anode, which makes a positively charged, positively potential lens, that is anode. These accelerated primary electrons are made to be incident on the sample through what means through a condensing lens and scanning coil. These high speed primary electrons on falling over the sample produces low energy secondary electrons. The collection on the secondary electrons are very difficult, and hence a high voltage is applied to the collector, because high that secondary electron what we are seeing, that is scattered from the specimen, so it is moving with a high voltage or with high velocity. So due to that reason, so we are applying so high voltage for to collect the secondary electrons. This collected electron produces a photo of multiplier tube or converted into an electrical signal. These signals are amplified by the video amplifier and is fed to the cathode ray oscilloscope. By similar procedure, the electron beam scans from left to right, and the whole picture of the sample is obtained in the cathode ray screen, that is cathode ray oscilloscope screen. So that means compared to the transmitted electron beam, so in this scanning electron microscope, so here what we are extra we are using means that is scanning coil. So why we are using this scanning coil means where we are kept this scanning coil means in between the condenser lens and the that specimen. So now see here, so this scanning coil which collects and into detector, that detector will detect that secondary coils. So that secondary coil will convert that into an electrical signal. So and it will generate some signal that it will send to the cathode ray oscilloscope. So this is the picture, this is the working part of the scanning electron microscope. So here it will explain external structure of the small sized materials, and main the difficult what we are seeing in this SEM means so that is scanning electron microscope means so that is the in the preparation of this small sized sample, that is only difficult to make or to, because in this scanning electron microscope that sample what we are using, that is in very small size. To make that very small size material, that is only difficult in this scanning electron microscope. So after this, let us see any operators if we consider, if we derive, so that all operators which has some advantages also and some disadvantages also. So like that, let us in this microscope also it has some advantages and also some disadvantages. Let us see in the next slide, that is the advantages of TEM, that is transmission electron microscope. So the main advantage of this high resolution is electron microscope is, that is high resolution magnified image we are getting. And already I told, so it can give then the internal structure and the chemical composition can be collected in the specimen or in the sample. At a time it will give. And if I consider the advantage of scanning electron microscope, image can be directly viewed. So what is the specimen we are taken, that directly we can view through that CRO. So next, it has a large depth of focus. So that means I told you. So the focusing in this scanning electron microscope is very good compared to TEM. So next, some disadvantages of TEM means, so that is aberration due to lenses. So that is because, see here already I told you that in that TEM, so we are eliminating a diffracted beam. So that diffracted beam, that is X-ray beam, while you are eliminating to get a contrast image or to get a magnified image, so that is only that making that that lenses to eliminate the diffracted beam, that is only difficult in that TEM. Next, in that TEM there is no 3D image. We can see the 3D image only in SEM, not in TEM. So in the TEM, so we are using a 2D image. So we are getting a 2D image, where in a TEM. Next, if I consider the disadvantages of SEM, the resolution of image is very poor. So why means, I told you that, so compared to SEM, the TEM which can give high magnified contrast and internal structure, at a time it will give the complete image. But in the SEM, so it is just giving out that crystal outside the external structure of the specimen. The preparation of sample is very difficult, it is ridiculous. So this is the advantage and disadvantage of TEM and SEM. Now next, see the applications of TEM and SEM. So this TEM we are using in nanofield for the to find the internal structure of the nanomaterial and to get the 2D image of your biological cells, virus and bacteria, in studying the composition of paints and alloys. And we can use it in biology related fields like microbiology, etc. And if I consider the SEM, the specimens of large thickness can be verified in by using the scanning electron microscope, and it is used to get the 3D image. I told you in the last slide itself. So that is from the TEM we can get a 2D image, and from the SEM we can get a 3D image. So that's why, so to get the some small cells so image, we are using this SEM to find the structural composition of paper pulps and the ceramic materials like polymers, etc. So you to find out that we are using a SEM. So this is the electron microscope. So there some more electron microscopes are there, so like atomic force microscope. So all the microscope we are using to just to magnify to get the resolution high effective images. So this is the our video. So in the next video, so we are going to studying them another, so that we will study in the next video. So thank you for watching this video. Thank you very much.